Hello, medical intervention students. I'm going to go through the December 15th and 16th plan. In class, you're going to finish up the Superbugs lab. The Gold have already done this. I kind of pushed them ahead a day on Monday because they have one less period compared to the blue group. So you're going to finish by confirming the growth, confirming that you have resistance with that resistance has been given from E. coli strain 1 to E. coli strain 2. If we're not sure what I'm talking about here, we're talking about this lab activity. So we did our mix plates, right? And we have then put those plates, put a sample from the mix plate on, we will put that onto the streptomycin, an ampicillin plate, and a strep and an amp plate. And this is the one where you'll get to do your own design. The Gold group did that on Monday. Blue group will do it on Tuesday. And then we'll receive the results on Wednesday and Thursday. And basically, we're just looking for any sort of growth. So that's why you get to do your own design on this. So each person will get to do that. We talked the other day about mutation, inactivation, and efflux as the methods by which antibiotics resistance happens. Now I want to talk just a little bit about these mechanisms of gene transfer. So you have conjugation, transformation, and transduction. So if you go back to the video here, and I'll kind of skip through here. We'll first talk about conjugation. I'm just going to go through the names. So basically, it's where a plasmid is basically transferred from one to the other. Okay, and so here, if you look in the animation, we have bacterial cells. They have a plasmid. Well, they can generate a little bridge between the two, and that's called an apily or the pilus. And so that bridge allows a copy of the plasmid to get duplicated to then go to the next bacterium. So if this plasmid has a gene for antibiotic resistance and it gets copied, then that gene goes into here, into this cell. Now this bacterium will have antibiotic resistance because it received this plasmid and can produce the protein as coded for on this plasmid. So now you have two bacteria, and then all of their offspring will have this plasmid duplicated as well, and they will also have it. So you're now going to spread that even faster. And then we have, to skip ahead, transformation. Transformation is where naked DNA is transferred from one bacterium to another. And so it's usually in the process of consuming another bacterium. So you have two bacteria, there's the chromosome, okay? And what happens is this bacteria destroys another or eats another or breaks down, other bacteria dies, and now it's taking in this DNA. And it may incorporate that into its own DNA. And if it has a gene that's helpful, then that gene may have, if it's an antibiotic resistance gene, then that antibiotic resistance gene may get incorporated to its DNA. Whereas previously, maybe this bacteria was resistant and this was not. Now it picks up those resistance genes, maybe on these plasmids, and is able to incorporate them into their own DNA. Okay, so that's transformation. So conjugation and transformation are just one big difference is conjugation is going to be both are still living and they're going to conjugate together. They're going to conjugate. And versus transformation, you are transforming a living bacteria from parts of a dead bacteria. All right, and then finally we get to transduction. We'll talk about this in class too. And transduction is completely different from the other two because that involves a virus. All right, so virus or phage, bacteriophage. Okay, so here's a bacteriophage, this little virus right here, and what viruses or phages can basically go into bacteria and take over the cell processes. We know all too well about viruses these days, but it may pick up some of this bacterial DNA into the viral structure, and then when it goes to infect another host cell, it can inject or put that DNA into those cells. So now these cells are all getting that particular form of resistance because these viruses happen to pick it up. So those are really the three types of antibiotic resistance transfer. So in this case, you would be talking about, you have a plasmid going from one to the other. That would be conjugation. Here we have donor DNA or naked DNA. All right, it's going straight over, but it's not a plasmid. That's transformation. And here we have the phage or the virus. That would be transduction. All right, and then we'll look at our final results. You'll turn this in. This is due Wednesday, the 16th. And then we are going to work on an assessment in class. I'm making it due the 17th. I want you to work on this independently in class, individually in class, for kind of think of almost like a test to some extent. But I want you to think about summarizing this. So let me explain this a little bit more. So the 1, 2, 3 assessment, I'll have a paper copy of it in class, but you'll also have a digital copy if you'd rather type it. So you're going to write a conclusion using the CER or claim evidence reasoning method. The claim should be about which bacteria passes resistance to which. Your evidence should discuss each bacteria structure and location of the gene. So the claim is basically, did E. coli 1 give resistance to E. coli 2, or did E. coli 2 give resistance to E. coli 1? And then you're going to relate that to the structure and the location of the gene. So was it on the nucleoid? Was it on the plasmid? What was resistant to what? Also, and then your reasoning should discuss which method of gene transference must have occurred and how we know and how that connects back to the evidence. So was it conjugation, transformation, transduction? All right, so think about which way did it have to go in order for that to happen, in order for the bacterial transfer to take place. Assuming that it did, assuming that our plates here all were positive. All right. And then the other part of that, which I see this part being done in class more, whereas this part probably something you have to work on at home, is you're summing up all the things from this activity, make a graphic organizer relating the terms, all these terms you see here, the 3 types of gene transfer, the 3 types of antibiotic resistance methods, as well as some other terms like gene, plasmid, naked DNA, pilus, bacteriophage. So basically, you're summarizing those 3 processes and the 3 ways that bacteria become resistant to antibiotics. All right, so we'll work on that in class as well. And then additionally, so you have really 3 assignments due this week. The third is the 1, 2, 4 when antibiotics fail. Hopefully you've done a lot of this. You've done the perfect trial. Hopefully you've also done the random trial. You should have done that in class and graphed the 3 lines, the resistant, least resistant, and very resistant bacteria. While we graph those 3 lines, remember you need a red line for the very resistant, a yellow for the resistant, and a blue for the least resistant. And then finally, another claim evidence reasoning. Just coincidence that these 2 happen to be right next to each other. So we will do these throughout the year. But your claim should be about how doses taken or missed affected the various populations of bacteria. So missing doses caused what to happen with populations of bacteria, or taking all the doses caused what happened to the populations of bacteria. Now, where's your evidence for that? That would be discussing different strains. So then you're going to go back and discuss, well, when you took a dose, how did that affect the different strains of bacteria? When you did not take a dose, how did that affect the strains of bacteria? Okay. And then finally, your reasoning, okay, should relate to the evidence to the claim. So discussing how it backs it up. So for instance, on that, you might say, I've claimed that taking the medicine causes the resistant bacteria to increase, all right, the most resistant. Well, does my evidence support that? Well, it means that every time I took the medicine, that should mean that my population of my red or very resistant bacteria went up. And so from there, I'm just going to tie that in and explain, okay, because the very resistant bacteria increased every time I took medicine, that supports the idea that taking the medicine increases the resistant bacteria, which is not true. I'm just making that up. So, but go through and look at what the data tell you, and then this is basically where you're spelling it out. So these are more facts, like here's what happened. Here's some evidence to support my claim, and here's your explanation of what actually did happen. Kind of in a paragraph or bullet points where you're going to give more detail. And so where do we turn this stuff in? In Canvas. Make sure you've turned in the 122 assignment. That should have been turned in last week. The antibiotic disc susceptibility tests. Superbugs lab data. You should turn that in class when you're here this week on Tuesday or Wednesday. I'll give you time to turn that in. The Superbugs assessment. All right. That'll be due Thursday as well. And the Superbugs assessment does count as an exam grade. So it's in that more heavily weighted category. And then the when antibiotics fail, graphing and simulation lab. So that is also due on Thursday of this week. Thursday, the blue group, I'm going to, and I'll probably give this out to the gold people anyway. We're going to start looking at, since your last day is Wednesday, I'll give this to you on Wednesday so that you can look at the next lesson where we're going to be talking about Sue Smith. And I'll explain that a little bit more on Thursday, but basically we'll start looking into 1.3 and it relates to hearing because people who suffer from meningitis can suffer some permanent hearing loss. So we'll talk more about that. So that's it. Please let me know if you have any questions, and I'll see you in class this week. Thank you.